McGill as a postdoc and until now as a faculty. And currently, Koren is like a department chair and full professor that is a pharmacology and therapeutic McGill University. And Koren's research is like a mito related to the toxicity and a particular cardiovascular disease. And the mito is arsenic and tungsten. And she has like a first grant in 2009 and leading as the PI and later and have several fund and grant support in Canada, the several foundation, Canadian Institute and Health Research and the National Science Engineer Research Council of Canada, Leukemia and Lymphoma Society, a lot of society, and also from the NIH grant, NIH support. And the current research and is related to mito and like a cardiovascular disease, and already published more than 75 peer review papers and training a lot of graduate students and postdoc, and they are very successful. But this is a very general and can read the word. But I want to say something and special. And yeah, and I think that like the word right now is very funny. Yeah. So if you see the word is big word and it's big word, and small word is a small word. I want to give you two cases and a small word. Like I just mentioned that Koren and went to Canada from United States to Canada in the 1999. And I got Canada citizenship and moved to United States in 1999. We are just, and then Koren go to McGill. I got my postdoc in McGill.
Why don't you try it?
Well,
say you guys got the better deal.
Yeah, they have a lot of the like special like, but that is just focus me and with Koren. Yeah. And like I did my postdoc in 1995 in the McGill and the same department, and my mentor still there as first faculty right now. And that is a McIntyre building, and she is in the top in the office in the 13th floor. And like our like a lab in the first floor, right? Still first floor, yeah. Yeah, and then later we are working the same specialty in SOT, mito special session, and she's president and treasurer. And so this is really a small world. We have a couple of the meeting on Zoom and online, but I see it's a big world, and I give you another the case. Like a pandemic really changed our life. When we have the meeting and talk with on the Zoom and like in mito like a special meeting also in the on Zoom. But one day we have a chance in Germany for the meeting. That moment they required you need to wear a mask. So when we go to the same like a meeting room, only three row and separate. When they finish and stand up, she hello, I said, oh hi. Have we met before? And she said, oh yeah, and move the mask. Oh, I realized, oh, it's Korean. And so that's a really big word. And even we have more than 10, 20 times met on Zoom without mask. And we know each other. And but when we really see the person, we didn't recognize. So that's why we have to invite her in person to come here and do another chance. We met each other. Thanks.
So now Lou can't forget me. The other thing that we did that you didn't talk about is we in Quebec had a pretty, I would say, strict COVID policy. They shut the province down. There was no one on the streets. We had to have special permission to go to the hospital, and my lab is at the hospital that's the primary COVID point, or was in Quebec. So thus we've all had COVID several times in my lab. But one of the things that they did was they shut us out, shut our labs down. They said, and it was for months. And about a couple weeks in, I'm meeting with all the students on Zoom, and you can tell that they're struggling. And I'm like, all right, so what do we do to fix this? So I called up Lou and John and I'm like, all right, can we have a group lab meeting or a journal club? Because they need to see other people and they need to be engaged still. And we did that all through the pandemic. And then we asked them, you know, they're going back to the lab. Adoya's back there. She's like, yes. Do you want to keep doing this? And they're like, yeah, we want to keep doing this, because what in the end happened was that they all showed up at SOT last year already knowing each other. They already had this amazing group of colleagues that they felt comfortable with and were talking. So yeah, we kept it going. Although this week I heard there was a revolt. We'll talk about that later. If I paid? Oh, I believe I did that last year in San Diego. Good to know. Nashville is coming up. All right, so I'm going to tell you just briefly what the lab is about. And I realize, and I apologize that this slide is crazy busy. Everything in my lab, so I trained as an immunotoxicologist. So one correction is that my PhD is actually while I was in a joint program. Am I standing too close? Yeah, I don't know what that goes. It's a joint program with BU and Harvard, but my degree actually says BU on it, so Boston University. I don't claim Harvard. I apologize for anyone who does. So everything in my lab, at least at the beginning, started out having to do with how a metal affects the immune system to come to some pathology. And at the beginning, I wasn't too fussy about what that pathology is. So if you look at my CV, I was telling Alexandra this yesterday, I look wicked diffuse. Like I'm all over the place, but I swear there's a theme. And that has to do with how metals affect the immune system. But that means that there's this really broad effect space or causative agent that causes lots of changes. So in the lab, there's two main focus projects that the big grants fund. That's on tungsten, which is sort of an emerging metal, on bone and how that affects hematopoiesis, particularly B cells and generation of leukemia. And I could talk about that if you want. I'm going to talk about the arsenic and atherosclerosis today, and I tried to make it sort of a focus for Chris. I tried really hard. We'll see. But we look at macrophages and the macrophage biology behind atherosclerosis and how that is modulated by arsenic. In addition, I guess as full disclosure, I also have a Victor grant with Jamie Melliker and Birgit Klaus-Henn, where we're looking at how arsenic and cadmium at low dose is linked to atherosclerosis, and I have to get Matt involved at some point in that one because we see a liver phenotype there. And we're matching it to the human cohort in Denmark. And then I'm also involved in the Columbia University Northern Plains Superfund, where I am co-PI on Project 4, which is using mouse modeling to look at combinations of arsenic and uranium. In addition, I don't have the ability to say no to people who ask and come in with really exciting things. So I have a project that's funded with Susan Gaskin to look at exchangeable copper levels in ALS with Bernard Robert, who is the former mentor of Lou, has asked me to look at some of the immune changes in his giant rat study that's transgenerational, the OP exposure. We have a pretty big project now on vaping and atherosclerosis using the AAV PCSK9 model. And then because I am at a hospital and the person with the office next to me is a hematology oncologist who specializes in lymphoma, and stepped in my office one day and said, hey, I want to collect all these samples from relapse refractory lymphoma patients. Can we do something? Like, well, you have to be more specific, but probably. So what we did was develop the first set of SOPs for collecting collecting biospecimens from multi-center clinical trials in lymphoma and how to process them so that you can actually use them. So again, I'm happy to talk about any of these with anyone, but I'm going to focus on athero today. So this is largely not needed in this room, but just to remind you that arsenic is epidemiologically linked to atherosclerosis, which is the buildup of this fatty plaque here in the large vessels, in the large arteries. People don't die of atherosclerosis normally. It does cause a narrowing of the lumen, but mostly what happens is you get pieces of that plaque that break off and go and clog something important, like, you know, the vessels feeding the brain or the vessels feeding the heart. So you end up with stroke or MI. And we know that arsenic is a risk factor. And then I have to say that this project is largely built on data that came out of Sanjay and Chris's lab, where they were one of the first people to publish arsenic in the apolipoprotein E knockout mouse. And so, yeah, I stole that and continued on. I just asked different questions, but I have to say it's been very useful and pleasant over the many years to have people to go and talk to and say, okay, this is what we found this week. What do you think, Sanjay? So I thank you for that. Just a reminder that atherosclerosis is an inflammatory disease. So again, there's a rhyme to my reason in this. That the inflammatory disease is really because of the retention of lipids in the subendothelial cell space. They become oxidized. This causes a recruitment of monocytes from the circulation. They transmigrate into the subendothelial space, differentiate oftentimes into macrophages, although there are macs already sitting there. They gobble up all that lipid and become foam cells. And this generation of foam cells goes on to cause a larger inflammatory response. That involves T cells, involves dendritic cells, B cells. As the plaque increases, what you get is the activation of this smooth muscle cell layer. And what happens is that they migrate above the fatty streak or above the beginning plaque. They proliferate and they secrete extracellular matrix proteins like collagen. And that's an effort to stabilize the, I knew this is my problem, stabilize the plaque right here. So the bigger the cap you have, so the more smooth muscle cells, the more collagen, the more stable it is, and the less likely it is to break off and go and do something horrible. So we borrowed Sanjay and Chris's model, where we started also our mice at 5 weeks of age here. And we use our sort of standard protocol now is 13 weeks of sodium arsenite in the drinking water. We replenish every other day, Monday, Wednesday, Friday. And then our endpoints, keep in mind, we put everyone on this horribly expensive purified AIN diet that is certified lower metals. We keep measuring it. FYI, it's not always in the little spec sheet. So it's not horribly different, but sometimes there are differences. And so our endpoints tend to be plaque quantification. So we look first here at the sort of gross plaque in the lumen of the vessel in what we call en face staining with oil red O. And we also look at the plaque in the aortic sinus. And anyone feel free to stop me as we go along if you have questions. The other thing we do is we look at what the plaque is made of. We look at macrophage content. We look at the amount of lipid within the plaque, the amount of collagen. Oh, I apologize for the typo. And smooth muscle cells. And that's sort of the standard protocol that we use. And we've used this model to show that even really low concentrations of arsenic will increase plaque. So this is 10 parts per billion to 200 parts per billion. And you see more or less a dose response in both the total en face staining as well as in the sinus. But one of the things that we characterized in this model and others, and other people have shown this as well, is that in the plaque, you get more fat. So inside the plaque, the amount of plaque, the amount of lipid is increased by arsenic. And this is normalized to plaque area. But we see absolutely no change in the number of macrophages in the plaque. None. So that sort of has to mean if those macrophages are holding on to the fat, that each individual macrophage has to be holding on to more. There are some other hypotheses, but that's our favorite one. So we came up with the hypothesis that maybe arsenic is not necessarily changing the number of macrophages, but the function of them. And the one thing you need to know about macrophages is they come in a bunch of different flavors. Because what these cells do are respond to the microenvironment. So they take cues from where they're sitting and they say, okay, what we need here is something that's going to act like a vacuum and clean up things, or we need a pro-inflammatory response. And they're very plastic, and the same cell can be one and then switch to the other and back again. So in atherosclerosis, at least when we started this, there were sort of a continuum of polarization, but in vitro, there was sort of the ends of the spectrum. And that included what used to be called M1 on one side and M2 on the other side. No one likes that nomenclature anymore. But the general idea is that these pro-inflammatory Th1 type cells are pro-atherogenic and they secrete lots of pro-atherogenic cytokines. And the other end of the spectrum are sort of wound healing, pro-resolving. And so almost all the work I'm going to show you today is by one of the PhD students that's going to defend soon in April. And Koren Mann is her name. And she and I put together this hypothesis that if you look in a dish, we can take bone marrow and make bone marrow derived macrophages. That takes about 5 days of MCS cell culture. And then we can push them to either be M1-like or M2-like, or we can just leave them in MCSF and they're what we call unpolarized or M0. And our hypothesis was that arsenic would come along and then they would all be M-arsenic because they're all sitting in the same microenvironment, so that you would have, everything would be skewed towards the center. Like most of my hypotheses, I was wrong. We did this experiment where we took bone marrow derived macrophages, differentiated them for 5 days to M0, and then put them either in interferon gamma and made them M1-like, or IL-4 and made them M2-like, plus or minus 50 parts per billion arsenic for 48 hours. And then we did bulk RNA sequencing. And what you can see is that we didn't move them at all. And in fact, we didn't even move them towards each other. So red is the arsenic treated, blue is the control group. M0s were pretty far as the PCA plot, but M1 and M2 are close to each other, but they certainly didn't move towards each other, suggesting that arsenic is affecting what started as the same cell in different ways. So if we do a little deeper dive into this, what we see is that the arsenic-induced differential gene expression in M1s look like this, and the IL-4 in the M2s look like this. And there's really very little overlap. And then even if you look at those overlap cells, the genes overlap, but their directions don't. So that all of these up here are increased in IL-4 stimulated, and all of these down here are increased in interferon gamma stimulated. And really, the only ones that overlap are these tiny proportion here, which is kind of cool because, again, we started out with the same cell, and arsenic is causing them to do completely different things. If we go a little deeper into what's changed, if you look at things that go in the same direction in the overlap, it's not surprising you find things like hemoxygenase 1. It's well known to be responsive to arsenic, and we see that too. Both, oops, excuse me, let me go backwards. Both on the protein level or the RNA level, and this is a completely different set of RNAs from the different set of cells. But this is 50 parts per billion, which ends up, I did the conversion yesterday. It's like 0.94. This, and I threw this up for you, Chris, because that's the HO1 plot. And we see differential splicing, we think. It is published in this differential splicing. And what you see is that there's a difference at basal levels just between the macrophages. But arsenic actually looks like it increases this smaller variant. So I give that to you and do what you want with it. Yeah.
Only because I'm Chris's technician.
Okay.
Do you know what kind of alternative splicing we've done?
Zero clue.
So,
I give it to you, and you are more than welcome to have all of that data. I think that's fascinating, and I don't know what it means here. It's just an observation that I made. Yeah. Some of the other things we found, so not everything went up, some things went down. And this, because I'm an immunologist, was intriguing to me. These are all MHC class 2 molecules. And by RNA levels, they go down in both of them. However, when we looked at the protein levels, and particularly the cell surface level of them, it's well known that M1-like cells have more than M2. So that's a comparison between here and here. But what you see is that if you treat them with arsenic, you lose these high expressors over here. We don't know what that means immunologically yet, but they do have less MHC class 2 on their surface. So then if you go and look at what's happening individually, like what is it doing to each of those cell types separately? When you put it through pathway analysis, Karen comes racing into the office going, are you never going to believe this? It's like I did the mirror image. I put the pathway analysis down on the table and put a mirror and said, okay, if you just move everything in the opposite direction, that's what happened. So what you see is that in the interferon gamma stimulated macrophages, you get an increase in the super pathway of cholesterol biosynthesis. In the IL-4, that's this dark blue, and then the individual pathways are here. And so if you look at the pathway itself, the ones that are down arrows in sort of this pale pink are the IL-4 stimulated arsenic changes, and then the green ones, like here, IDI1, go up. So they're exactly opposite, which is really intriguing since they started in the same cell. One thing I'll just point out is that we were very happy to see that in the interferon gamma stimulated LXR RXR activation, so that's liver X receptor and retinoic acid receptor, because we started this with data showing that arsenic inhibits LXR, and in fact inhibits in macrophages any nuclear receptor that heterodimerizes with RXR. So it sort of validated the previous 10 years of my life. And we went on in the paper, there's some bone marrow transplant studies that you can go look at and show that that's still important. But this doesn't actually help us to try and understand what is sitting at the plaque. That's all in a dish. So everyone kept asking, well, how relevant is this? Because there have been several studies now looking at single cell RNA-seq data from the plaque in both mice, like the ApoE knockout mouse, the LDL receptor knockout mouse, and looking at what types of macrophages and immune cells in general are sitting there. And almost across the board, you end up with 3 types of macrophages when you do these single cell analysis. One is a resident-like macrophage that looks like a cell that was probably sitting there before and wasn't necessarily recruited. One here is the inflammatory macrophage. Some papers call them the foam cell phenotype, that were probably the ones that were monocytes and were recruited. And then there's always this other. In this particular paper, they call it TREM2 high. Everyone sort of has their favorite marker, and they call it that high macrophage. We do too. So if those are what's sitting in the plaque, we wanted to know what arsenic did to those macrophages. So we also jumped into single cell RNA-seq. And for the first very expensive experiment, we decided to hedge our bets. So we used 5-week-old male mice, 4 controls and 4 arsenic. And we put them on a high-fat diet to make sure that the plaques were going to be nice and big. And then either gave them tap water control or 200 parts per billion for 13 weeks. And then we added a step in here, and that is to control for circulating immune cells. Because we were interested in the cells that were in the plaque, not those that were in the general circulation. So about 5 minutes before sacrifice, we inject a BB515 labeled anti-CD45 antibody that will label all of the cells in the periphery. 5 minutes is enough to basically label everything. Sacrifice, cardiac puncture, perfuse, harvest the plaque, digest it, all the ECMs, get the cells out, and then we stain again. We stain then with the BV785 labeled anti-CD45. So same clone, different color. And then we sort based on those cells that are BV785 positive, so plaque resident, but BB515 negative, so not from the periphery. And then we use the 10x genomics platform. What else was I going to tell you about that? I guess that's it. So initially, I will present here actually multiple slides on the computational analysis. I have an amazing computational biologist that I work with. And while I may attempt to answer any questions, I will most likely refer you to June because my knowledge and understanding of his computational magic is a service only. Which is exactly opposite of what we tell all of your students to do. Don't put anything on a slide that you can't explain, but I'm going to do it anyway. So the first thing we did was just the clustering analysis, and this is both the control and arsenic. So again, pooled from 4 mice each. And in this initial attempt, we got a total of about 2,500 cells. So I think we did fair. You'll see data later on that we've gotten much better at this, and now we routinely get about 5,000 cells from that. And the clustering gives us about 19 different clusters. When we overlay the cell types using a couple different data sets on immune cells, including MGen, the mouse data sets, and also just cherry picking, looking at the data and saying, okay, well, that's a CD3 cell, it's got to be a T cell. You end up with a set of T cells over here. These are granulocytes. This is sort of our unknown population. These tiny little set here is B cells. These are dendritic cells, and then all the purple are macrophages. And if we look, zoom in on those macrophages, we pick up the same 3 cell types. So you see that the resident-like cells are over here. These are the foam-like cells or the inflammatory ones. And then TREM2 didn't come up with us, so we couldn't name them TREM2 positive. But the interesting gene that turns them on is THPS1. So we call these THPS1 oxides macrophages. So that led us to some, I guess I was happy to see that data because it didn't mean we threw that money down the toilet. We can actually see the same kinds of cell types that other people see. And then if you overlay the exposure on top, what you see is that here, the orange are control and the blue are arsenic. And there are some clusters that are more arsenic and some that are more control. But if you go back to the sort of total cell type number, there's no big change in the types of cells that are sitting in the plaque, which sort of, again, matches our staining, particularly with the macrophage data, which is in the green. Here you go to 60 to 65%. Maybe you get a little bit fewer granulocytes B cells, but it's not a massive shift of the immune population in the plaque. All right. But if we go back to the original clustering, what we see is that there are some clusters, the granulocyte cluster is the largest, and it's profoundly enriched in control. We haven't followed this up yet, but it's certainly a low-hanging fruit, probably. But there are other clusters that are pretty enriched in arsenic. This is normalized by number of cells in the cluster. And we went on to do an RNA velocity analysis. And briefly, my understanding of RNA velocity analysis, uses the three prime splicing analysis of the reads and uses whether or not you've gone from unspliced and spliced to infer a pseudotime or a trajectory. And so I'll point out just a couple of these. So if we look at the resident-like macrophage clusters, which are 1, 10, and 4, 1 sort of seems to come out of nowhere and we don't really understand that well. So focus yourself on 10 and 4. 10 is a control cluster and 4 is an arsenic-enriched cluster, so ABC. In the foam cells, the control enriched cluster is 2, but it makes not just one cluster, but 3, 8, 13, 7. So it increases the heterogeneity of the foam cell-like. And so when you look at what pathways are involved, and we've just focused for simplicity's sake on some of the larger clusters, so 10 and 4 as examples of resident-like, and 2 and 3 as examples of foam cell-like. And if you look at the differential gene expression that's here, if you focus again on the fact that there's very few common changes between the two. So that matches our in vitro data. So Koren's like, well, we have to know if they're moving in the same direction then. And in fact, they don't. If you look at just those that are upregulated of the 162, in the middle, that's the overlap there. So 20 some genes that are both upregulated and 8 that are downregulated. So that's the sum total that go in the same direction of your 162 genes. Not very many. Most of them go in opposite directions too. And you see that when you go into the pathway analysis. So this is the resonant-like cluster comparison between 10 and 4. This is the foam cell-like cluster comparison between 3 and 2. And you can see that IL-17 signaling goes up in the resident-like, but down in the foam cell-like. And the same is true for chemokine, cytokine receptor signaling. They go in opposite directions. Also, TNF-alpha signaling is here and here. So we're seeing the same thing in the plaque that we saw in the dish. Okay, that's good. So far, what do we know? We know that arsenic doesn't really change the total component of immune cells, that macrophages still are the largest component number of cells in the plaque, whether or not there's arsenic there or not. And they are the most modified cells by arsenic. T cells are not particularly changed. B cell population is too small to know anything. The dendritic cells show a little bit of change, but not in any magnitude the same as the macrophages. Interestingly, arsenic increases the heterogeneity of the population of foam cells. I'm not sure what that means yet, but it's an interesting observation. And that we see sort of the opposite trends in gene expression regulation in the two types of macrophages, comparing foam and resident cells. It's a lot of data and a lot of observations. I'm not sure I got anywhere close to helping myself answer a mechanistic question. What is arsenic doing in the macrophages that's making the plaque bigger? So, like every good computational biologist who doesn't have the lab, June says, oh, we need to do single cell multiome. Here, it's only $30,000. And when Koren says, yeah, we should do that, I'm like, all right, here you go. So that's what we did. And so single cell multiome in this case is both a repeat of the single cell RNA-seq, but also single cell attack-seq from those same cells. So you're looking basically where the chromatin is open or closed. It's about chromatin accessibility in the attack-seq. So nicely, the single cell RNA-seq looks very similar in the multiome analysis to our first single cell RNA-seq, again, 13 different clusters. In this graph, the original cluster that we can map it back to in the first data set is in parentheses. So here are the resident cells, resident-like macrophages in this sort of, I don't know, ugly green color. And that was 10 before, and here it moved to arsenic as cluster 4. They're now called 5 and 6, just to be confusing, but we try and annotate it as closely as possible. Again, We found no difference in the real change of the number of the different immune cell populations that are there, the percentages. We do find all the same similar clusters. Here, this is the example we went. So 2,600 cells total the first time we did it, and we got better at it from the same 4 and 4 mice. So now we get double the amount of cells just by practice. We're better at getting the plaque out. But the same types of cells are there more or less. Then if you move to the single cell attack-seq data, you get a similar number of clusters here. This is the clustering. And these are the cell types. But what you'll notice is that, like, if you look at this sort of greeny teal color, there's a set of resident-like macrophages sitting on the left side of this bump. And then there's a set here. They're no longer sort of right next to each other, and they don't move from one to another that we can tell yet. But what happened is when you overlaid the exposure, all of a sudden it was like a dream and completely separated by the left side being control and the right side being arsenic. It's not 100%. You see some orange cells here, there, here, but for the most part, it's pretty obvious that arsenic is transcriptionally driving the polarization or the changes in the macrophages. Whether that means there's some epigenetic change, DNA methylation, whether that's a transcription factor recruitment, activating a certain set of transcription factors, we don't know yet. But what we've done, and more importantly, what June has done, is look at genes that are arsenic enriched and those that are control in different clusters. And if you overlay the transcription factor motifs, right? This is not transcription factor expression. It's not chip data, so I don't know what's sitting on that piece of DNA. It's just the DNA motifs that are open in the case of attack-seq from the multiome, and then looking at target genes that are enriched for these same or the transcription factors that are enriched in the target genes. This is the initial single cell RNA-seq, and this is the single cell RNA-seq from the multiome. And what you see is that we have this pretty still long list of transcription factors that are enriched, or at least their binding sites are enriched in the attack-seq and also in the single cell. So the question then becomes, which ones of these are important? There are many of them that have flocks mice available so that we can go in and look at the importance in different knocking it out of macrophages, maybe driven a little bit in vitro first, since while we don't find M1 and M2 in the plaque, we do seem to see similar arsenic-induced changes there. So the question still becomes always, how relevant is your mouse modeling to human exposure? So there's a data set that was published in Nature Medicine in 2019 that is from human plaque. And what they looked at was plaque from symptomatic and asymptomatic people. So on the top is our macrophage data, so mouse macrophage, and just focus again on 10 and 4, those clusters of resident-like cells. You can find similar clusters in the human data set, actually, clusters 7 and 6. And we match this, just showing you that we can find the same markers in the macrophages. But when you compare the data sets, cluster 10, which is a control cluster, looks a lot like cluster 6. And this is enriched in asymptomatic patients, p-values associated here. And cluster 4, which is arsenic enriched, looks a lot like cluster 7, which is enriched in symptomatic patients. So it's possible that what arsenic is doing is switching the macrophage phenotype towards the one that we see in symptomatic patients, which would be bad. So that is sort of our comparison with mouse and human. I threw this in for the meeting, as well as some other people that asked where we're going with this. because it's still a ton of data and I'm not yet to mechanism. That's a long list of transcription factors to dig through. Is there a way we can narrow things down a bit? And so, of course, Jim's like, oh yes, we can do that, no problem. All you need to do is throw several $100,000 more at this, not a big deal. Yeah. So here's the plan. I know this is incredibly small, but I did that on purpose, so you don't ask me too many questions. But basically what we're gonna do is some exposures where we have plaque increase and plaque decrease. And we're gonna do this by assessing at 13 weeks, 17 weeks, and 21 weeks. Assuming that the plaque is gonna get bigger, sometimes more complex, but we'll be able to tell this. We're gonna compare males and females because in mice, it's opposite of humans. Females get bigger plaques than males. And we're gonna compare diet, which is either a control, just the regular AIN diet, and a high-fat diet. So all of those things, we should be able to look at things that make the plaque worse, so up. But in the machine learning and the AI, it's important to also have some idea of what might make the plaque smaller. It makes the prediction more robust. And so we're going to add in one, we don't actually know that this is going to make it smaller, but that's the prediction, is the washout. What if we put them on arsenic and then we take the arsenic away? The prediction is that the plaque will get smaller. We don't actually know that yet, but we'll have a set of mice that we assess that at the same time. The data that we do know, however, in a very odd collaboration, was with a group in Saskatchewan that was interested in looking at selenium-enriched diets. Weirdly, the pulse board, pulse meaning like lentils, the Pulse Board of Saskatchewan has a lot of money, and they were willing to pay us to test whether or not selenium-rich lentils from Saskatchewan would inhibit plaque formation in our model. So we created the diet, we had a low selenium diet, we had low selenium lentils from I don't remember where, and turns out they're not wrong. So the high selenium lentil diet actually protected against plaque formation. So in theory, we can use this as our regression model if on the off chance the wash off doesn't work. So the idea is to plug this all into the machine learning model and then have an idea of what nodes are important. And maybe we can narrow down that list a bit so that we get at some of the mechanistic questions. We already have some ideas that osteopontin and CD44 and integrins are important. Based on some cell chat analysis, looking at receptor ligand pairs in the macrophages. But I think when we get this all done, it's going to be a much more robust data set that can hopefully lead us somewhere. But the one thing that we're already starting on is how we're going to validate what we're finding. Like there's a lot of data. It's all RNA or DNA in case of the attack-seq. We all know that really it's just the proteins that matter. So how are we going to validate this? And what we acquired through the grace of some donors at the hospital was a Codex phenocycler system. I don't know how many people are familiar with the system, but basically it's a way of highly multiplexing immunofluorescence. So we haven't done it at this level, but some people have used 100 different markers. We've done 30 plex as our top number so far. And the way it works is that you use bar-coded primary antibodies. You stain the tissue slide with all of your primary antibodies. And then you have 3 different secondaries, the corresponding barcodes, 3 different colors, and Dept. And you stain with those first 3 images. And then because the nucleic acid interactions are so labile compared to protein interactions, you can just wash them. And then you come in with another 3 antibodies with different barcodes and you're done. And you just keep doing this in rounds until you get done. And what it allows you to do is the Dept allows you to overlay all the images so that you're in the same plane and the same space. And then you look at all of the colors at the same time. So this is a lymph node that has a tumor in it. So it started out and was developed for immune system and cancer. That's where most of the data have been done. And to my knowledge, no one's used this with atherosclerotic plaque, but you know, that's never stopped us before. So what I want you to do is, this is all tumor in the lymph node, but focus down here is sort of the residual normal germinal center of this lymph node. And then if we blow it up, it looks like this. Blue is DAPI, or dark blue is DAPI. The T cells are in this lighter blue, CD11B, 19 are the B cells, Ki67 proliferation marker, and this is alpha pseudoslapid. And you can zoom in a little bit farther and you see nice, like if you look at this cell, I'm going to move the pointer so you can actually see it. You see nice F4 staining on the periphery. And you can take the data then and use it like a UMAP, or in this case we used Tisney, to sort of map all the different cell types. And then you can overlay that information back onto the original anatomical location. You can ask nearest neighbor questions. You can ask over time. The algorithm is quite robust. The data files are humongous. We have now our own server for these, and it's quite powerful, but the problem is we didn't know if it would work in plaque. So we just started this. And normally when we do plaque, we do it in frozen tissue, the OCT tissue. But you need to do formalin fixed here, really. So it was a whole experience trying to get the slicing through the aortic sinus. But we managed it. What you can see here is in green is the smooth muscle. So that looks very nice and pretty. And in the blow-ups on the top, what you see is CD206, which is a marker of endothelial macrophages. And on the bottom, it's CD45. We just tried it, and the reason there's two images is because we had barcodes for those, but they were the same color, and we just didn't want to have to reconjugate the antibodies to do the test. So that's why there's two images. In theory, when we do this for real, everything will have some color. But we can take then the markers that we see that separate out clusters by single cell and then go in and validate. You get not only protein, but you get spatial anatomical information by doing it this way. Yeah, we're still in the process of that, but that's the plan. We'll have to obviously do, people always ask me this, well, that's not very many cells. Like here, if you look at the CD45 just in this little corner of the plaque, it's not very many. I don't know how much information we can get from that. But the thing is, you can image a full slide, and we can put 12 slices of plaque on a slide. So we're just going to have to image more to get a full picture. But I think all of that is doable because in the end, if we stain it all at the same time, we can overlay the UMAP or Tisney data and map it back onto each individual slice and still do nearest neighbors. And if we know this sequence of the slicing, we can actually even then ask between adjacent slices nearest neighbor questions. So that is where we are. This was my lab in summer when everyone first got to take off their masks. So that's why some of them look so happy. So this is Koren. She did the bulk of this work. She's really an amazing PhD student. She came actually to the University of Arizona on a Fulbright from Pakistan, and then decided that she wanted to be in Canada. So Clark Lance actually hooked her up with us, and I've been thankful. This is June. He's the computational biologist, and his student Shui. This is the funding for this particular project, both from the Heart and Stroke Foundation of Canada and Canadian Institute of Health Research. Christian Young is my flow cytometrist, and Claudia Clement and Nicholas DeJay helped us with the bioinformatics from the bulk RNA-seq. Happy to take any questions. Go ahead, ask away.
Okay, thank you. I just had a question. Have you ever considered actually looking at the cell, like the wheel cell? And if the macrophage numbers are changing and the types are changing dramatically, does it suggest to me that there's some outside component, especially if there's more extending component that would suggest stronger, more robust plaque, so I wondered if you investigated your genes for it.
Yes, we have. So my bias, to be honest, is to focus on the immune system.
Yeah.
But we have looked at that because we know from a lot of lineage tracing studies that smooth muscle cells actually can differentiate, right? And take on